Is this now? Okay. So good morning, everybody. Welcome to our first of this semester's four research talks by faculty at the university. The intent, of course, from the perspective of the students who are in the class, is to make notes, make sure you are up to date with whatever we may ask you about these talks during the exam. If you're not here for class and you're just interested, that's the right thing to do. And please come back for the following talks as well. So today we will have Dr. Matthew Kirk from our Department of Geology here at Kansas State. He just described himself to me in words that I would not have come up with, so I'll parrot those to you. He is a geochemist, right? Geochemist, who acknowledges the fact that microbes exist, that life exists even within rocks, and that life to a large extent drives the chemistry in rocks and in the soil. He has a huge background also in groundwater, which is interesting from a Kansas perspective since we are very dependent on our groundwater, especially in the West. He has an enormous reputation. He has 27 internationally peer-reviewed publications out there for all of us to read. And they've by now been cited more than 600 times, which means that a lot of people find his work useful and interesting. So we're very happy to have you here today, Matt, and I'm going to give you the proverbial microphone, although you have your own, I know. So with that, I'll give the floor to you.
All right, thank you. Is this on? I can't, sounds just like I'm talking normally right now. Okay, well, can I turn this light off over here? There we go. There we go. All right. Well, thank you very much, Arno, for the kind words and the introduction and the opportunity to come meet with you all. It's a pleasure to share some of this stuff with you guys today. I know that to some extent it's a captive audience, but I do appreciate that you're here. So I'm excited to talk about some of our work that we've been doing that examines anaerobic microorganisms. And kind of one of my main goals for this presentation is to just help you understand why anaerobes are important in the environment, why they play important roles in environmental chemistry, and why that's useful to know about for environmental science. So I'm going to start out talking about my motivation for this research. I'll share with you some results from bioreactor experiments and some applications of the knowledge that we've gained, and then I'll summarize our main conclusions. So, I am a parent. This is my son, Emerson. He's going to be 2 in December. And, I've always been interested in environmental quality issues, but since entering parenthood, I would say that my motivation for this area of research has definitely heightened. I'm genuinely worried about the world that we are handing our young people and the world that we ourselves are going to inhabit, and that we already inhabit, obviously. This has definitely increased my motivation to contribute to solving some of these big challenges that we have. Parenthood changes you in a lot of ways. But in any case, I'll stay on track here. One of the things that I'm concerned about, and I suspect many or most or all of you are concerned about, is climate change, anthropogenic climate change. The UN report from the Intergovernmental Panel on Climate Change that came out, I think about a year and a half ago, that was associated with this goal of limiting the warming of Earth to 1.5 degrees above pre-industrial levels. That report had a lot of numbers in it that were summarizing a lot of science from people around the world. And those numbers are very troubling. So we have this goal of limiting warming to 1.5 degrees. And if we can only emit about another 420 gigatons of CO2 before we have enough CO2 in the atmosphere to achieve that level of warming. It isn't necessarily going to happen immediately, but we'll have enough CO2 for that to happen. And based upon the rate at which we are emitting CO2 currently, and the fact that it's increasing, by the time my son is about 10 or maybe in middle school, we're going to have enough CO2 in the atmosphere for 1.5 degrees of warming. And that's troubling, right? If we go over 1.5 degrees, we're not going to fall off a cliff, right? We're not going to just perish immediately in a ball of flames. But we're at a degree of warming now, right? And it's not good. We already have impacts at a degree of warming. 1.5 degrees is worse. 2 degrees is worse than that, and so forth, right? So the longer this thing goes on, the worse it's going to get. It's going to cost us one way or the other, and we can be proactive about it, and we can start to reduce some of those costs and do it in a smart way. So that's something that I'm very motivated to try to help with. Another UN report, this one from the Biodiversity Panel, came out about a year ago, and it concluded that about a million species on the planet are threatened with extinction right now. Okay? It's because of climate change, but also because of pollution, land use, impacts, things that we do, to the land, including, loss of habitat, exploitation of critters. Now, this is not a good thing, right? These aren't just cuddly creatures that we think are nice and we want to, see running around in the wild. These are organisms that contribute to our economies, right? And also provide ecosystem services that help make life on Earth possible. So this is a big concern as well, right? And it's linked to climate change. We need to start taking these things seriously. If we're not going to do it for ourselves, let's do it for our kids, right? So I'm very motivated to contribute to solving these problems. And one of the ways that I can do that is through my research. So in my research group, what we do is we examine interactions of microorganisms with geological environments. And kind of the key motivator, the main drivers of our research are to better understand factors that control water quality and to better understand carbon budgets in soils and aquatic habitats and places like that. So we have a project that has been going for a few years now, where we've been looking at the impacts of land use on the chemistry and microbiology of a portion of the High Plains Aquifer and the overlying soils. And that's in south central Kansas. It's a portion of the High Plains Aquifer known as the Great Bend Prairie Aquifer. And we should have our first article from that research coming out later this year. We also have a big project funded through the Kansas EPSCoR program that is examining microbiomes of aquatic habitats, plants, and soils, and how those microbiomes interact and how they're influenced by the precipitation gradient that exists in Kansas and land use superimposed on that precipitation gradient. So this is a big project. I'm just a small piece of this project. It includes a lot of people at K-State, people at KU, Fort Hays State, and Wichita State. Some really great scientists collaborating from broad perspectives on this research. I'm really grateful to be a part of that. My student Stefan has taken a closer look at some soil redox chemistry, literally and figuratively, and how that relates to other physical and chemical parameters in these soils across the state. And that's going to help us learn about carbon budgets and how much carbon is stored in these soils. My student Bree is studying variation in surface water groundwater interactions across the state, and different drainage areas across the aridity gradient that have different land use proportions, let's say. Ultimately, we want to know how the amount of groundwater discharge into these stream environments is influencing stream biogeochemistry. And the work that I'm going to focus on today is mainly looking at environmental controls on groups of anaerobic microorganisms. And the current project that we have is being carried out by Javil, who's one of my graduate students. It's funded by the National Science Foundation Geobiology program, and specifically we're looking at environmental drivers of interspecies electron transfer between a couple of groups of microbes. I'm going to talk more about interspecies electron transfer later on because it's pretty cool, and I hope you guys agree when I don't bore you to death. All right, so let's talk about those bioreactors. When getting into this stuff, I think with an audience from different backgrounds, I think it's useful to kind of bear in mind and remind ourselves what respiration is and how our own bodies work, right? So we oxidize organic carbon that we get out of the food that we eat, and we reduce oxygen from the air that we breathe. So in other words, we are taking electrons away from organic carbon and we're putting them onto oxygen. So we can say that organic carbon is our electron donor and oxygen is our electron acceptor. I'm going to use those terms a lot. Electron donor, electron acceptor. Well, there's a lot of microbes that do the same thing. They respire oxygen. Oxygen is the preferred electron acceptor for life on Earth, so it's pretty common. It's a nice thing to use as your electron acceptor. But in the absence of that, microbes can use a lot of other things as their electron acceptor. They can, and this is in anoxic environments, right? So they can use nitrate as their electron acceptor. They can use ferric iron, the iron that's in iron oxides and oxyhydroxides. They can respire sulfate, and they can also respire CO2, and that's one of the ways that they make methane, a microbial reaction known as methanogenesis. All right, so I'm going to focus on these three microbial reactions down here at the bottom. Iron reduction, sulfate reduction, methanogenesis for the rest of this talk. These are three of the coolest microbial reactions that exist as far as I'm concerned. I am a little bit biased, but I think the chemistry is really interesting, and I hope you guys like it too. So I became interested in these microbes as a master's student when I studied arsenic in the Mahomet Aquifer. So the Mahomet Aquifer is this glacial aquifer system in Illinois. It's basically a bunch of glacial sediments sitting in a bedrock valley that got filled in when the glaciers came cruising by. So the water is really good. It's really tasty water. But in places there's arsenic in the water, which isn't great. Arsenic's not good for your health. So when I was doing my master's, we decided to look into this and try to understand a little bit more about what microbes are doing that could be helping influence, the mobility of the arsenic. And what we observed is that arsenic levels were only elevated at hazardous levels in places that had very little sulfate, where the water was virtually free of sulfate. And we also noticed that high arsenic water tended to have a lot of methane in there. So we used these data, as well as some other data that we had collected, to start to put together a story about how microbes were influencing the chemistry of the aquifer and how that affected arsenic. And so this is what it looks like. So one of the most important controls on arsenic mobility in an aquifer is adsorption. Arsenic likes to stick onto iron oxides and oxyhydroxides. When iron-reducing microbes start using those minerals as their electron acceptor, it causes the mineral to reductively dissolve. The mineral dissolves. So any arsenic that it contains can get kicked out into the solution. It becomes mobilized. If microbes are also respiring the arsenic, they can also mobilize that arsenic because they're changing the oxidation state of the arsenic and affecting its ability to sorb. So this seems to be happening throughout the aquifer, arsenic being released into the groundwater. But in places you have sulfate, and where that sulfate is present, you have microbial sulfate reduction, which produces sulfide that can react with the ferrous iron produced by iron reduction and form sulfide minerals. Probably starts out as the mineral macanoite, FeS, and then transforms into the mineral pyrite over time. And pyrite's pretty good at taking up arsenic. And that seems to be what has happened in this culture that's about a dozen years old. If you look at it and shine a light in there, you can see crystal faces. so that pyrite has started to form in this old culture as a result of sulfate reduction. So sulfate reduction, if that's happening, and that seems to be happening wherever you got sulfate in this aquifer, you have a sink for arsenic. And it seems to be in the Muhammad aquifer and many other reducing aquifers worldwide, if you have that sink, arsenic isn't a problem. If you take that sink away, arsenic can accumulate and become hazardous to humans who consume the water. We also see that if you don't have sulfate and you don't have sulfate reduction, you see methane start to accumulate in the water alongside that arsenic because the sulfate reducers can't outcompete methanogens for electron donors for the food. If those sulfate reducers don't have sulfate, they can't do sulfate reduction. That means there's some leftover food in the system that the methanogens can eat and turn into methane. So that was our interpretation. It was mostly based on chemistry, but also some molecular microbiology. Subsequently, people have done more detailed microbial investigations that also are consistent with this model. So that's what kind of introduced me to these groups of microbes: methanogens, sulfate reducers, iron reducers. And thinking about environmental chemistry and how microbes and environmental chemistry co-evolve and what that can mean for things like water quality. So I was hooked. I thought that was really cool. Over the last 10 years or so, I've kind of kicked the arsenic habit and started thinking more about just environmental controls on the balance between these microbial reactions in nature. What are some of the ways that the environment influences the balance between these processes? So initially, people thought that these microbial reactions were kind of distributed into discrete zones in places like aquifers or marine sediment. And there is some truth to that, but we know that that model is an oversimplification. People figured that out pretty quickly. And one of the reasons, there's a lot of reasons, but one of the reasons is because iron reduction and sulfate reduction often occur simultaneously. And we also see that iron reduction and methanogenesis also can occur simultaneously in places where sulfate isn't available. So the reason seems to be that iron reduction is a slippery rung on the thermodynamic ladder. It isn't always a really great thing for microbes to use. Sometimes it's really nice, sometimes it's not so nice. So that causes it to kind of overlap to varying degrees in terms of its use as an electron acceptor with these other microbial reactions. The reason why it's kind of more variable than these other reactions, one of them is the fact that unlike sulfate reduction and methanogenesis, iron reduction is a heterogeneous reaction. It includes the aqueous phase as well as a solid phase. Right? So microbes are trying to transfer electrons to ferric iron in that solid. One of the things that can ultimately serve as a bottleneck on the rate of that electron transfer is the surface area of that mineral. Right? The oxide surface area is a limitation on the rate of iron reduction. So if electron donor is being supplied at a rate that starts to push on that bottleneck, food can get diverted to alternative microbial strategies, even if iron reduction is extremely favorable otherwise. Right? And so those alternative strategies can be sulfate reduction or methanogenesis, which are both, they're not involving a solid phase. They don't have that surface area limitation. Another thing that affects iron reduction that doesn't affect sulfate reduction and methanogenesis is sorption. So I talked about sorption before with regard to arsenic, but the product of iron reduction is Fe2+, ferrous iron. It can also sorb onto metal oxides and oxyhydroxides. So iron reducers take ferric iron, which is largely insoluble at near neutral pHs, and turn it into ferrous iron, which is mostly soluble, but some of it can stick back onto that oxide. Well, the extent to which that happens is pH dependent. All right, so this is a really simple reaction path model that includes some goethite and a little bit of ferrous iron, and then I've just slid the pH from 5 to 9 and looked at how the concentration of sorbed ferrous iron species changes. And you can see that at pH 5 and 6, there isn't a whole lot of sorption going on, but as pH increases, that changes. So what this tells us is that sorption probably becomes more important at basic pH than it is at acidic pH as a control on the rate of iron reduction. All right. Similarly, the amount of energy that's available for a microbe using iron reduction also varies with pH. Right? So iron reduction, I'll show you a reaction later, but iron reduction consumes a lot of hydrogen ions. What that means is that as pH increases, or in other words, the activity of hydrogen ions increases as pH decreases, the reaction becomes much more energetically favorable. So, and that's what I've plotted here. This is energy available for iron reduction, sulfate reduction, and methanogenesis relative to pH. And again, this is just from a simple sliding pH reaction path model. And I've done iron reduction for a few different iron phases. So this one is goethite, which I'll talk a lot about today. And what you can see is that at acidic pH, iron reduction generally has an energy advantage over sulfate reduction and methanogenesis. But at basic pH, sulfate reduction and methanogenesis can be more favorable than iron reduction, depending on what source of ferric iron is present, what mineral is present. All right, so let's try to put all this together before we move forward. We have two kind of main working hypotheses that we've been playing around with for the past few years. One of them is that iron reduction is going to be better able to compete with sulfate reduction and methanogenesis at acidic pH than basic pH because of this variation in energy and because of sorption. The other one is iron reduction, the amount of iron reduction relative to sulfate reduction and methanogenesis. We think that it would decrease as you increase the amount of food because of that surface area limitation on the rate of iron reduction. So two kind of main hypotheses. All right, so we set up these bioreactors, and basically they consist of serum bottles, just like this one right here. Serum bottles that are partially filled with some aqueous medium, some water. We add microbes by either adding marsh sediment to the reactors, which is what we've done most of the time. And for some experiments, we've also just injected them with groundwater that has microbes in there. Either way, you have a very diverse community of microbes that can do a lot of things, including iron reduction, sulfate reduction, methanogenesis. We added goethite to most of the reactors to provide a source of ferric iron for iron-reducing microbes. And then once a week, we would remove 1/5 of the aqueous volume of the reactors and then immediately replace it with fresh, unreacted medium that contained a little bit of acetate to serve as a food source. All right, so basically we're trying to roughly simulate what happens in an aquifer where you have minerals that are kind of fixed in place and water flowing through, right? So this is a kind of a crude way to simulate that. The water that we would take out every week, we would use that for a bunch of chemical analyses. One of those is we would measure the amount of ferrous iron in that fluid, and that helps us understand how much iron reduction is happening. So this plot shows ferrous iron concentrations over time, reactors that received medium with pH 6, some that received medium with pH 7.5 water, and then you can also see over here that we varied the availability of sulfate and the availability of acetate. Notice this one doesn't have any acetate. We still have a lot of biological activity because that marsh sediment had a lot of organic matter, which is food for microbes. So a couple of reasons why I'm showing you this graph. I don't want to get too far into the weeds here. First one is that I want to point out our variables. pH was one of our variables in our experiments, availability of sulfate, availability of acetate, and also the availability of ferric iron. Another thing I wanted to point out was just how we use these data. So we would take these wet water chemistry results, plug them into some mass balance calculations, combine them with some sediment extraction data, and that allows us to calculate how much iron reduction occurred during the incubation. So we would do the same kind of thing for sulfate reduction, similar thing for methanogenesis. And when we put those results together, we can start to understand how the balance between these microbial reactions change as we tweak these, environmental control knobs. Right? We can understand how the balance, the proportions change. So let's take a look at what that looks like. Right? So this plot shows the reactors that had sulfate. And so in these reactors, the main thing going on was iron reduction and sulfate reduction. And on the y-axis here, I've ratioed the amount of iron reduced to the amount of sulfate reduced. And then x-axis, I have pH. And one thing you can see is that at acidic pH, we have more iron reduction than sulfate reduction, consistent with our hypothesis. We also see that the proportion of iron reduction relative to sulfate reduction decreases as we increase the amount of food. And we think that's because of that surface area bottleneck. At basic pH, we see that the amount of iron reduction almost always matches the amount of sulfate reduction, regardless of how much acetate is flowing through that reactor. So that's kind of a cool result. We've seen that now in several experiments. And we think that there's a few reasons why that happens. So unlike acidic environments where iron reduction is very favorable, both in terms of energy, sorption, etc., at basic pH, iron reduction is kind of just barely scraping by. It's getting hit with sorption. It doesn't have a lot of energy available from its reaction. So it needs help, and sulfate reducers can provide some help. And one of the reasons we think sulfate reducers are providing help, one of the ways, is by causing iron sulfide minerals to form. So sulfate reduction produces sulfide, iron reduction produces ferrous iron. Those two things can precipitate with one another, form minerals like this that we talked about before. As a result, you've got less ferrous iron in the solution. So that means there's going to be less iron sorbing onto that mineral. Helps keep the mineral fresh and tasty for iron-reducing microbes. In addition to that, if you're limiting accumulation of ferrous iron in the fluid, that helps preserve the energy yield to some extent of that reaction. It helps limit product inhibition. Another idea that's out there, our collaborator Ted published, and there's some other similar studies as well, but the idea is that sulfate reduction helps iron reduction happen by triggering some sulfur cycling that indirectly drives iron reduction. So let's break this down a little bit. We got sulfate reduction producing sulfide. That sulfide abiologically reduces iron. That seems to be more favorable than microbial iron reduction at basic pH. And it produces sulfur species with intermediate oxidation states, which I've represented as elemental sulfur there. Well, that elemental sulfur can then be used by iron-reducing microbes as their electron acceptor, replenishing the sulfide that can then, help drive iron reduction. So the iron reducers are driving iron reduction indirectly by helping cycle sulfur. And so it might be that they switch to that strategy as pH increases because iron reduction becomes, biological iron reduction becomes less favorable. So this is a pretty cool idea. Like I said, it's out there in a few other studies. But one thing that's important to know is that I'm still calling these critters iron reducers. But with the microbial world, just realize that there's a lot of metabolic flexibility, right? We can respire oxygen. Well, iron reducers can respire iron, but they can respire other things as well, right? That's pretty cool. So always bear that in mind. That's why I've got iron reducer in quotes. So these are two of the ways that iron reduction can persist in environments that have basic pH and have sulfate. Let's take a look at environments that don't have sulfate. Here we have interactions of iron-reducing microbes and methanogens. And again, we see that the proportion of iron reduction relative to methanogenesis, it drops as pH increases. Again, that's consistent with our hypothesis about pH. Interestingly, we see an opposite result when it comes to the amount of electron donor that's available. I don't know exactly why that is. You know, it's hard to pin down, but we would have to do more work to figure it out. Part of it might just be because methanogens are relatively, their kinetics are pretty slow. And so relative to the time scale of the experiment, even though there was more food available, they weren't able to kind of take advantage of that within the incubation period. But we need to do some more work to kind of figure out why that is. Either way, it's interesting. It's the opposite result that we see in environments with sulfate. Now, one of the things that we think is kind of underpinning this drop in iron reduction as pH increases is a switch in the way that iron reducers, and methanogens interact with each other. All right, so at acidic pH, iron reducers are really happy. They've got everything going for them, right? The reaction's yielding a lot of energy. There's not much sorption going on. And so they're very happy to eat food and transfer the electrons to iron oxides, produce ferrous iron. And effectively, they're competing with methanogens for that food source. But what we think happens as pH increases is that they increasingly start to participate in interspecies electron transfer. And in this case, they eat the food, and instead of transferring the electrons to the iron oxide, they instead hand them off to the methanogen, puts them onto CO2, and makes methane. If it occurs through nanowires or some direct connection, it's called direct interspecies electron transfer, or DIET. And you know, this is pretty cool. It's a form of syntrophy, but basically, you know, it's another option for those iron-reducing microbes. If iron isn't a great electron acceptor for them, they can collaborate with those methanogens, and essentially they're participating in methanogenesis. They're still able to grow a population, but they're not actually reducing iron, either directly or indirectly. So if we look at the microbial community directly, we can start to learn a little bit more about what's going on behind the scenes. The chemistry tells you kind of the net result, what's happening at the end of the day. But some of the cycling going on behind the scenes can be preserved in the microbial community. So we analyze the microbes in the reactors by extracting their DNA, amplifying it with polymerase chain reaction, PCR, and then sequencing that DNA and doing a taxonomic analysis to say, you know, what categories do they fit into, which groups are present. And then we can take those results and you know, say, well, which of these groups is at least capable of iron reduction? Can't prove that that's what they're actually doing, but which ones are capable of that? Right? And we compared that to the amount of iron reduction that happened. And you see, these are just best fit lines. Obviously, it looks pretty good when you have, you know, just two ends of data. But the thing is, you see a direct relationship between the amount of iron reduction and the amount of the relative abundance of iron reducers in the microbial community in experiments with sulfate. In experiments without sulfate, we do not see that relationship. Even though there wasn't much iron reduction going on down here in these reactors, those were the reactors that had basic pH, we still have a pretty high abundance of iron reducers, much higher than the amount that we added, implying that there was some growth. And the reason we think that they were able to grow that community without reducing iron, we think it's because they were doing interspecies electron transfer. So that's what Javil is testing out right now. So we'll see if that ends up being correct. But right now we have some circumstantial evidence that suggests that that's the case. All right, so how can we use these results? Well, I'm going to kind of go through this a little bit quickly because I think I want to make sure that we have some time for questions if there are any. So, if you have questions about it, make a note. But I will say that the simplest way we can use these kinds of results is just, interpreting environmental chemistry. That's something that we actually do. So this is just comparing our results to variation in the chemistry of natural aquifers across the U.S. And we see that we do have we have consistent relationships there, and that helps us understand why we see variation in, for example, the amount of iron and sulfide in water, and the amount of iron and methane as a function of pH. So we can start to interpret environmental chemistry based upon, in a more informed way, let's say. Another option for using these kinds of results is to kind of use them to improve process-based models for methane generation. So methane, very potent greenhouse gas, obviously. We can estimate how much methane is forming with a top-down approach using satellites. There's also these bottom-up approaches, process-based models that take into account, the conditions that are present on the land and try to estimate how much methane is going to be generated. They're important if you want to try to project into the future and estimate how climate change, for example, is going to affect methane emissions. But it turns out those process-based models don't always see eye to eye with those top-down approaches, right? So there's some inaccuracies. And maybe if we incorporate the interactions between iron reducers and methanogens and how they change with pH, we might be able to better describe these carbon budgets, the carbon budgets of these systems. So iron reduction has a very different impact on carbon budgets than methanogenesis. Instead of making methane, it helps trap CO2 by consuming hydrogen ions. So that's a really big number there. And if we want to see how that affects CO2, we can combine it with this reaction that describes carbonate equilibrium. We see that per mole of acetate, 15 moles of CO2 are converted into bicarbonate. So you're taking inorganic carbon from a form that can degas into the atmosphere, and you're putting it in an ionic form that's more easily retained in an aqueous system. It can also precipitate as a carbonate mineral. So it's good at trapping carbon. Methanogenesis is good at making a potent greenhouse gas. So if we account for, if we're better able to account for the proportions of these things in process-based models, we'll be better able to predict how those carbon budgets will change. Another thing that we can do with these kinds of results is develop biotechnology. We can use these environmental control knobs to manage biogeochemical processes in both natural and engineered systems. So some of the basic environmental control knobs on microbial populations include pH, temperature, salinity, availability of energy resources, nutrients, water, fluid flow. We've been talking about how we can adjust pH and affect the balance between two microbial reactions, right? That's potentially useful. One example would be arsenic bioremediation. One of the implications of this was that we could simply resupply sulfate to these areas that have high arsenic, stimulate sulfate reduction, cause arsenic to be removed from the water. This is really great. Compared to building a water treatment facility, which isn't really an option in less developed countries, pumping in some sulfate would be pretty simple. This has actually been tested out since then and shown to be a viable option. It's a lot more complicated than what I'm describing to you right now, but it does work. Another example is energy production. We can use those control knobs and energy strategies to harvest energy from microbes. So the most obvious example would be methanogenesis makes methane, which is the main component of natural gas. If you capture that methane, it's good. If it goes to the atmosphere, it's bad, right? So you could potentially adjust the amount of methane forming in a system based upon environmental control knobs, and that could be good if you're getting the methane back. You could be getting methane that has modern carbon. Another example is microbial fuel cell. This is a microbial fuel cell that is available. You can buy it on Amazon. And that's not a plug for Amazon. That's just saying you can go get one of these if you want. This company, Magical Microbes, makes these things called mudwats, and we made some with students this summer. Here's one. Basically, things like iron reducers, they can transfer electrons to iron minerals or microbes. They can also transfer electrons to electrodes. And there's also microbes that can harvest electrons from electrodes. And if you put those two things together, you get electron flow. You get electricity. And so that, there's potential strategies for energy production that involve some of the stuff I've been talking about. So very briefly, our conclusions. Anaerobic microbes play major roles in the environment. We've been talking about water quality and carbon budgets. There's certainly more to it than that. Knowledge of environmental controls can help us refine predictions of biogeochemical feedbacks and develop biotechnology. At its simplest level, we can also use these types of experiments to interpret trends that we observe in natural systems. And then we can help solve environmental issues in many ways, right? So we can do it through our work or research. We can do it by making better personal choices, in terms of how we make and consume, or how we consume energy and water. Making people aware of things, and also, working to get policymakers and other leaders elected that acknowledge the reality of the environmental challenges that we face. So I know that I'm speaking to the choir here, but I hope you guys are energized to go out and help make contributions to these problems. So thank you very much. If you want to come look at these things at the end, you can. Do we have any questions? Meredy.
Do methanotrophs have Do methanotrophs have any role in what you're looking at, or are you just mainly focusing on the methanogens?
That is a really good question. So methanotrophs are microbes that eat methane. And if it weren't for methanotrophs, we probably would all be in hot water, right? The Earth would have a lot more methane in the atmosphere. There's huge amounts of methane that form, for example, in ocean sediment. And if it weren't for microbes eating that methane before it gets to the atmosphere, we would be done. But the work that we've been doing has largely focused on the anoxic environments where methanotrophs aren't playing a big role. There are methanotrophs that can combine methanotrophy with iron reduction, but they didn't take their opportunity to grow in our experiments. So, good question. Do we have any biology majors in this room? Is anybody biology? Chemistry. Are you guys K-State students? Where? I'm just kind of curious what departments? Shout it out. Geography? Animal science?
Biological systems engineering.
Okay, same. Okay.
Agronomy.
Agronomy. So these microbes, there's anaerobes in our guts, right? So we're talking about like large scale, like methane production in cows, and certainly lots of applications in BAE for learning about anaerobes. Have you guys been exposed to some of this stuff in your courses already a little bit? That's good. Well, if you haven't, I hope I inspired you to, learn a little bit more about them.
I'd have one more question.
Sure.
Yeah. So now you should get it too. Okay. I loved how you were describing how you look at the chemistry and then try to tease out what that means for the microbial activity and their interactions between different microbes. And I think there are beautiful metaphors right around the corner that you're no doubt aware of, with different restaurants and people moving to restaurants and so on. It made me think when you made that link to improving process models, is how do you tease out the functional forms? Are relations linear or are they exponential or logarithmic? Like, are there good ways that you use in your discipline to do that?
I guess, that's a really good question. I don't know if I'm really able to answer it very well. I would say that I wouldn't assume a linear relationship in most cases. Maybe it's best if we assume a nonlinear relationship. And we can look at data like this, right? We have statistically significant correlations there, and these are based on rank correlation tests, right? So they don't assume linearity. And I think for iron reduction and sulfate reduction, it probably isn't. It's probably something like this. But how do you account for that? I don't know. I think it's probably going to be a big data question. Yeah, and these data. This is from like the National Water Information System has 400,000 sites or something like that, and multiple samples for some of those sites. So there's a lot of data, but there's not a lot of methane data, and there's not a lot of sulfide data. So at the end of the day, I don't have that many data points there, even though we went through, a bunch of aquifers, right, across the U.S. And so part of it is going to be we need more data. You need data. So, big data. All right, well, if there are no more questions, thank you all for your attention. It was a pleasure.